It's a real pleasure to introduce our speaker this month, Dominique Durand, a colleague and friend. Dominique is in the Biomedical Engineering Department here at Case, where he's the Lindseth professor, and he's also a professor of Neurosciences, Physiology, and Biophysics, and he's the director of the Neural Engineering Center. So Dominique received his bachelor's degree from the École Nationale Supérieure in Toulouse, France, and MS from here at Case, and PhD from University of Toronto. He's very active, as many of you probably know. He's a fellow of the IEEE and of AIMB, and a fellow of the Institute of Physics. He's on a number of editorial boards. He's the editor of the Journal of Neural Engineering, and he has a broad range of research interests, some of which we'll hear about today, but not all. Does a lot of work in computational neuroscience, the neurophysiology of epilepsy, nonlinear dynamics, and a number of other things. Today, he's going to talk about peripheral nerve recordings. So, without further ado, Dominique.
Thank you very much.
You can,
oh, right.
Unless you want to walk around.
Well, thanks very much for inviting me. I don't know how many of you know this, but I've been here for a long, long time, 1982, actually. But my interests in neuroprosthetics, I didn't start with interest in neuroprosthetics. When I started here, I came from a lab in Toronto where we were doing electrophysiology, mostly brain slices, and mostly hippocampus. But as I got recruited to work in this department, and watching my colleagues, such as Tom Mortimer, he's not here today, I got very interested in this field. And in particular, as you know, Tom Mortimer at the time was doing a lot of work with the spiral nerve electrodes. And I was doing my brain slices, and I was looking at him doing his work, and noticed that sometimes these spirals that go around the nerve actually found their way into the nerve. And the end of it would just sort of penetrate the nerve. And based on that idea, I started to get interested in this, thinking perhaps we could put electrodes inside nerve, between fascicles, to record and stimulate. And I started a research project in the summer with summer students working on this. And then Dustin joined the lab, and then we come up with a bunch of different ideas to interface the nervous system with electrodes. And so what I'm going to talk to you today about is how do you interface the peripheral nervous system with electrodes. And I will show you different ways of doing this, and I also try to show you the progress that's been made in the last 10 years, 15 years. I've been working on this. See how far we've gone in this field and what we think we can do with it. So here's some of the ways that people have tried to interface with the peripheral nervous system. And you would probably think about the fact that it shouldn't be that difficult because we have clear access to a nerve. This is a peripheral nerve. And all the axons are right there inside. Why is it so difficult to connect with these axons, get signals, stimulate, and then build some prosthetic devices that are actually functional? Well, it seems easy, but it isn't. And you can see that a lot of people have tried different ways. And this electrode right here from Huntington in LA is the only commercially available electrode right now on the market for nerve peripheral interface. This electrode has been modified by a company called Cyberonics, and they stick it around this nerve here, and they use it for control of seizures in epilepsy on the vagus nerve. Then everything else you see is experimental and prototype, no commercial available. This is the Sieved ID, which I think was a great idea, still may be a great idea, but hasn't found a lot of applications. Basically, the idea is to cut the nerve and then have them regrow inside these holes there. And then since there's an interface with this, then you can obtain signals or stimulate and be very selective about it. Unfortunately, axons don't really like to go through small holes by themselves. they like to go through holes with their friends, and they like to form fascicles. And turns out that they are quite large, and they don't go through this too easily. So something else new has to be done, like maybe some micro machine device. Or this is the electrode from Utah. This is a 100 by 100 array, and that electrode is placed inside the nerve and penetrates the epinerium that you see right there. Right, it penetrates the perinerium, which surrounds the fascicles, and go right into this nerve. Now that device has been tested in animals, and a different version of it's been tested in brains. Of course, it's used everywhere. But the problem with it, I think, is that it penetrates this membrane, the perinerium. That membrane here is tight cell junctions, and it maintains a high pressure inside the fascicle. There's about 30 centimeter water inside there, and it also acts as a nerve-brain barrier. So it's an important barrier that may not be so easy to penetrate without causing damage. However, once you have these electrodes in there, they can be selective. They can be very selective and pick out signals from individual axons or individual nerves. They can stimulate selectively. So if this could be made to work, I think that could be a good technique. But the problem is avoiding potential damage. This is a spiral electrode that Tom Mortimer, who just worked in, has pioneered and developed for a long time. And Dustin Tyler has actually put this electrode in humans with pretty good success. So that electrode has gone a long way, all the way to the clinic. This device here, also from Utah, is a different idea. The idea is, can we put some wires inside the nerves and have them de-insulated at some points? And you can see right here, there's a de-insulation. So you take a needle, basically sew it into the nerve, drag this wire behind it, and leave it in there. And you have some spots where you are recording neurons, and activity. Problem is you don't really know where it is, where you are. And you're also going through the perinerium. Now this technique has been updated now with polyimide deposited film, and it's quite sophisticated, and it's quite popular in Europe. And there's several groups using this device. And then I want to talk to you about this right here, which is the fine flattening interface nerve electrode. And the shape of it is very different. It's flatter, and we put contacts on the inside, and I'll tell you why in a second. So the idea is this: if we were to, by the way, I'm skipping a lot of work that were done to get to this point. This is Dustin and I thought that this was probably one of the good ideas to pursue, because if we could maximize the circumference of this electrode, we would be able to place a lot more electrodes that around this, and then build a better interface with these fascicles. And you can see here, because a round electrode is probably the least optimum device for nerve interfacing, since the circumference is the smallest for a given cross-sectional area. And you can see here these two shape. One is circular, has an area of 0.7, and they have the same cross-sectional area, but this one has a much larger circumference than this one. So in principle, the idea is sound. If we could line up all these fascicles one by one, we should be able to put electrodes everywhere up and down here and be able to interface effectively. So here's some examples of this. Here's an electrode. You can see it has a bunch of fascicles right there. This is one that was built by Paul Yu, who's now at Duke University. And this one is designed for recording. You can see it has a very flat shape. It has 4, 5, 6, 7 electrodes here, and then one reference on each side, which is typical for recording. So with these devices, you can do recording and stimulation. We haven't done it simultaneously, but it could be done potentially that way. So we originally got a lot of sort of flack for this, because surgeons would say, well, this is a very nice idea, but I'm not going to let you take a cuff electrode that's flat and make my nerves flat. I'm not going to do this to my patients. Well, it turns out that nerves are not round anyway. Nerves tend out to be flat in the first place. Not always, not everywhere, but in many cases they are. And Ken Gustafsson did some really interesting work mapping the fascicular diameter and versus the number of fascicles, and then also looked at the width of the nerve versus the height of the nerve for a bunch of different nerves. And you can see all of these here. There's a dog sciatic, rat sciatic, etc. So what you see on these two plots is quite interesting because, at least to me it was interesting, is that we were used to fascicles we see in dogs, small animals, and they are always about 3, 4, 5 fascicles, pretty small. And we thought that in humans or larger animals, it would be about the same picture, but bigger fascicles. The answer is not the case. Fascicles remain the same size, about a millimeter or less, but they increase in diameter, in number. You can see, for example, here, this one, this is the sciatic nerve of a calf. And that has 300 fascicles. A human might have like 20, 30 fascicles. And the dog or cat might have 4 or 5. So the idea is that there must be some kind of optimum fascicle diameter. It may be, the reason may be because of the optimization of the blood supply, perhaps, or some development rule that we don't know about. But basically, the idea is that in humans, there are quite a few, a lot more fascicles than we thought there were. Turns out that if we want then to study this in humans, it's not so easy because there aren't too many animal models that have this very large number of fascicles. In fact, none of the models that we have here do display these large numbers. The only one that I've found recently is pig. Pigs have a large number of fascicles. I don't have a picture of it there, but it's about 20. I'm talking large pigs. And I'm still working on piglets to see what they do, what they have. But this is a problem that we're facing, is find a good animal model for this process. What's also interesting is you see that the height of the nerve doesn't increase either. In fact, nerves flat, very large nerves, are no more than 4 millimeters in height, but can be quite wide. It can be up to 2, 4, 5 centimeters wide. So nerves tend to be shaped in a natural flat configuration, especially when they're stuck between fascia, muscle fascia. And they take this shape, but in the end, though, I think what happens is that this shape is very flexible and plastic. So a nerve, you find that this is femoral nerve, for example, this one's femoral nerve, that you find in a human might have be flat in one area and some other area might be rounded. So it depends where it's sitting. And so these nerves are quite plastic and can be reshaped pretty easily. I'm going to skip a lot of data on the safety of this electrode, which I don't have time to go through, but I want to show you 3 things. One is how to do selective stimulation, to do selective recording, and control of a joint with a fine electrode, and then finally, how could we envision a device that could restore function in amputees? So a brief couple of slides on selective stimulation. Selective stimulation is an area that we started to work with because it was easier it's easier to stimulate than it is to record. And so we started with that, and this is the work of Matt Schiffer and Dustin Tyler. And you can see a very sophisticated modeling of the femoral nerve in human. This is a cross-section of the nerve with all the different muscle groups associated with certain number of fascicles. So you can see this finos is one associated with 5 fascicles. Some other ones are only 3. Vastus medialis is only 3, etc. And they differ in size, but overall the size are about 1.2 millimeters or less. And so you can actually build a computer model of this and test the idea that I can
it's possible to activate some and not others. And that idea has been on the computer model, I think, very well done here, where you can see that if I look at this bar here, which gives you the recruitment benefit, which is how much of the fascicle that you activate contributes to the movement, and the recruitment cost would be how much it deters from it. If you subtract that, you have the net activation that is what you want. And you can see that for all these different muscles, these numbers are quite high, 0.6, 0.7, in a very tight group fascicle. So even in a very tight situation, it is possible to recruit selectively these fascicles, and in principle. So these are some examples of experimental demonstration of these, and this is the work of Dustin Tyler when he was in my lab at the time on the cat sciatic ankle joint force. And you can see that these are all the different fascicles, CP, LG, tibial, MG, etc. This is a cross-section of the nerve. You can see the fascicle being activated, and these are the contacts of the electrodes. And you can see it's possible to recruit selectively either the green one here with these contacts, or the black one here with these contacts, or the red one with these contacts. So clearly I can produce force in this direction, etc. And this has been done several times in different animals, and it's been done in cats, it's been done in dogs. This is an example of work in the hypoglossal branch by Paul Yu. There are three main branches there in the hypoglossal: medial branch, styloglossus, hypoglossus, and genoglossus. And there are, it's possible to select those selectively as well. So selectivity of stimulation is good, because it means that we should be able, in principle, to control a joint by selective stimulation, but also to restore sensation in patients with amputation. And work in that area is going on right now at the VA. So let me then show you the next step is, if we can indeed control or selectively activate certain fascicles, can we control a movement with a cuff electrode? So this is the work of Andrew Park, who is about finishing his PhD soon. So the idea is this: if I have an electrode here, a multi-contact electrode, and I place it on the nerve, can I activate these different fascicles and control the movement of this ankle joint? You would think the problem is not that easy, because this is what's called multiple input, multiple output controller. And that means I have many inputs here that map into multiple outputs. And because this map is not a one-to-one map, there is redundancy involved. So several configuration of stimulation could activate several the same configuration of muscle, and vice versa. So it's not trivial. And also you would think that, that's what I thought anyway, well, if I activate a contact and then another contact after that one during movement, right? That movement is going to be very jittery, because I'm pulsing it. I always think of electrode pulsing and activating muscle. You get twitches, so it's hard to think that the movement here would be smooth in any way. So we started to look at this, and the idea was, one problem we're trying to do first is to avoid redundancy. And that problem, and that is the number of muscles around the joint is larger than the number of degree of freedom of the joint. So therefore, a given motion, there will be different combination of muscles, and that is very tricky to go around. So there was one idea that was put forward in 1993 by a Japanese well-known control expert, Kawato. And his idea was to separate the controller into two components. One component is steady state component. So this box here takes care of the steady state properties of the controller. And another box controls the dynamics separately. And so there's a feed-forward controller that takes care of that, and there's a PID controller that takes care of that. So this separation helps with the problem of redundancy and also helps simplify the controller. So you don't have to put everything into one box at one time, try to control everything at one time. And so we picked up on this idea and modified it. He published one paper on it, but never really pushed it beyond the, some simulations. So Onjo Park picked up on this and built a computer model of this applied to the joint mechanics of the ankle, and then did some experiments to see if that could be implementable. So here's the model that he used. These are all the muscles that were implemented, MG, LG, tibia, TP, etc., that control the ankle joint. These are the inputs, and the outputs are the joint angular displacement. So here's the muscle excitation level. This is the dynamics, and you get the angular displacement on the outside, on the output. The muscle activation dynamics for each muscle is standard. We took that from literature, 97 from Rainier, so there's nothing new there. And so this was also tied to a computer model of the nerve with the many various fascicles and the various contacts. So what he did is actually build, I think, is the first model of a complete system that has the nerve, the cuff electrode, the multiple contacts, the selective activation, the connection to the muscles, and the joint dynamics all in one computer model. So by activating these contacts, it's possible to make that joint move. So the dynamics for the neural model was implemented on the artificial neural net, and we used also the model for the axon standard models with sodium channels, etc. The diameters were distributed uniformly in this range, so we placed randomly the nodal ramier. So the model of the nerve is quite accurate. And we placed different axons, different diameter randomly inside the fascicle, and we use Warman's equations for solving it. So now let's look at the different parts of the controller. This is a steady state component. So the way you would build that steady state controller, you would tell the computer that I want a particular joint angle, and you would find what stimulation and what electrode would produce this. And then we build an inverse map. So we can map in 2 dimensions, this 2D plot here, the dorsiflexion and the inversion. And you can see that it's possible to place a point here just about everywhere, but not everywhere. There are regions that cannot be mapped. Like for example here, there's no parameter possible to get there. And so what he did then is build interpolation. So everywhere that we want, if we want to go to this point, right, for example, we would do interpolation from all the points around there, and you can see how that's done right there. This is standard feedback controller, the PID controller. I don't want to get into that. And the feed-forward controller is a multi-layer perceptron. Standard controllers. And here's some of the results. So this is on the computer, right? Here's an example of a sinusoidal input. This is a Dizard, is in blue. And what the system does is in red. You can see it follows rather accurately. And this is a 2 degree of freedom system. So we're controlling both the dorsiflexion and the inversion with this system. What's interesting to note is to look at the contacts. What do they do? And to produce this movement, right? This is time on this scale, and this is current on this axis, and the different colors here are the different contacts. There's about 20 contacts in there that produce different current at different times. So the controller, to produce this movement, would pick out a contact, right? Would figure how much current to put in, turn it off, find another contact, turn it off, etc. So then this multiplicity of an addition of all the currents as a function of time would produce this movement. Interestingly enough, he was also able to add a component in there of coactivation. So the steady state map was built in such a way that you could have a map at a different coactivation level and pick out which level you wanted during the movement. So you can see right here, for example, it on purpose shifted the coactivation level by stepwise fashion here. It was able to produce more coactivation. So this is important because during a movement, the coactivation level changes, and that affects the stiffness of the movement. And so that might be an important component to be able to control as well. Here's another example with pseudo-random noise, and you can see again that the controller follows pretty well the desired trajectory here, in both areas. Here's a context again, but what's interesting is also here is to look at what the individual muscles do and to produce this. And you can see that every one is sort of active, except perhaps this particular one not very active. Most of them are active at some time during this movement because they receive different kinds of stimulation. And this summation of this movement produce this, of this muscle activity produce this movement. We did one big problem with these controllers is robustness, and it's not so clear how they'll be resistant to fatigue, and we still don't really know how to solve that problem. But the controller was somewhat able to handle it because if you decrease the maximum strength of the muscle, possible strength by 50%, the controller is still able to follow somewhat, not exactly as good as before, but somewhat what the desired trajectory are. So there is some hope that perhaps this can be handled to some extent. And then he tried to do this in an experiment, and the experiment is in a rabbit. And you can see that in this case, the ankle is tied to an arm there, and the ankle, the knee joint is held by a pin. So this ankle is free to move, and the electrode is placed on the sciatic nerve. And what we're trying to do here is control the movement of this ankle using the same controller. I'm going to try to see if that movie is running. So you can see the movement is controlled, and actually, surprisingly, it's smooth, sort of smooth, although we didn't really quantify that looking at second-order derivative. But it's somewhat smooth, and this is, I think, the first time we've been able to control movement using multiple control. And in fact, here, this can be done fairly simple. There were not that many contacts involved in this. One of the big problems still here is the extent of the movement. You can see that it's not fully extended. And in fact, over the course of the experiments, the amount of the total angle of displacement here was reduced over time during the experiments. And also, one of the big problems with this technique is over time, that steady state map had to be redone a few times. It's not very robust in that area, and that is something I'm not sure how to handle. Perhaps it's because the experiments are acute and the electrode is moving, or perhaps I don't think it's fatigue because we sort of control for that. But it's an important still advancement because clearly this can be done. Whether it can be pushed as far as a full control movement is not so clear. We were able to go about 1 hertz, 2 hertz on this, 1.5 hertz, I think, not more than that. So there are still some problems, but there is some promise there that this type of electrodes, multi-contact, can in fact control movement. Now let me show you. Why doesn't that work? You can't advance it? Maybe it crashed. Let's go to recording. Now that is a more difficult problem. So here I showed you already, I'm showing this again so you can see the difference between a recording electrode and a stimulating electrode. A recording electrode has one contact separated by two outside references, and this has to be of a certain length, which is not short. It's about 1.5, 2 centimeters long. If it's anything shorter than that, the signals are too small. And already they're pretty small to start off. So this has to be quite long, which puts some strain on the nerve, because if you have a long electrode, which is a little stiff, and a nerve at the end that's tipping over and bending, so that could cause some damage. Whereas a stimulating electrode, you don't really need this tripolar arrangement. You could just have one electrode and be done. And you don't need to have such length either. So then how do we record and test whether this device can selectively record activity? And the experiment is shown right here. Here's an electrode placed on the hypoglossal nerve. And for these experiments, we'll work with this nerve because at that time we were working on obstructive sleep apnea for a stimulation problem. And so we use the same preparation to test this idea. So you have three main fascicles. Each one has an electrode that is activating this nerve. And then we can therefore control the input in each fascicle. And we can determine whether each electrode here is picking up the right signals. So there again, we do use a combination of experiments and modeling. A lot of modeling is going on in this experiment. Here's an electrode right here, and you can see the different fascicles. In this case, instead of putting an electrode and putting current to activate these fascicles, we do the reverse. We place current sources that are traveling at the speed of an action potential inside these fascicles, and then we record the signals. Oh, thank you, man. We record the signals on these different contacts here. So when this fascicle is activated, then you will see signals picked up by these electrodes. So we reproduce fairly accurately the signals. Here's the EMG, the electroneurogram, is generated by the following. We take the shape of the current that comes out of a node or NVA extracellularly. It looks like this. And in the literature, people have showed that you can actually build an EMG signal by taking the sum of these randomly delayed. So if I take a large number, say 10,000 of these, and add a random delay to this, I produce a signal like this. That signal, this is on the computer, right, looks very much like this signal right there, and that is a neural signal obtained by Mr. Sahin when he was here on the hypoglossal nerve. So there is, in fact, since then we've done some additional work on this and look at the frequency spectrum of this signal compared to this signal, and they're very similar. So we're pretty confident that the signals that we generate here are sort of physiological. And then we place then these signals inside the nerve, right? And we model this volume conductor, we model the cuff electrode, we model these different contacts here. Here's the nerve right there. We place that signal either here, or there, or in the combination of them. And here's what we observe. So the first thing we wanted to do is there a difference at all, any difference between different contacts when you stimulate one fascicle or another? You have to realize up to now, right, most people, when they recorded ENG, were placing one single electrode around the nerve and recording, or a hook electrode or cuff electrode, recording from the whole nerve at once. Here, what we're trying to do is to record only for certain fascicles. So you can see that it was encouraging here because this fascicle right here, when it's active, produces at contact number 1 a signal, the large one here, right, which is much larger than when number 3 is active, because when number 3 is active, you get the little guy. But on contact number 3 right here, right, it's reversed. So clearly, the position of the fascicle with that signal makes a difference and generates a different signal at different contact. So this is encouraging. So we decided to come up with some kind of an index, a selectivity index that would determine how well these signals can be separated. And this signal index goes from 0 to 1. And we compare that index to experiments. So this is model, and this is experiment. You can see the fascicles are almost the same, right? Same distribution. And you can see the signals are also almost the same. These are not stochastic signals, these are evoked signals, right? And you can see that again, the number 3 is active. It's larger of this contact than the other one, so sort of similar result. And then we plotted the selectivity index, which is calculated by the distance, Euclidean distance between signals, and plotted that for both. And you can see that both the experiment and the modeling agree pretty well. And the selectivity index varies from about 0.2 to 0.6, 0.7, and depending on the distance between sources. So if the sources or the fascicles are separated by distances in the 1.5 millimeter range, right, which is this line right there, you can see I can get a selectivity index of about 0.5. It's true for both the model and the experiment. And so it suggests that yes, it's possible, especially since the fascicles are in that range, right, like 1 millimeter, 2 millimeters. So possibly these signals can be separated. And so it's nice to see that both the experiments and the modeling agreed on that. So it's one thing to say, okay, I can separate this fascicle. Yes, I can recognize they're different, right? But that's just the beginning, because what you really want is to recover the signal. I want to see what the signal is. So that's another problem right there. That is not so easy. So then that's the next piece of work that was done by Wonde Testayefus. He was here a while ago. He had to leave because he got a job working for a company, which actually competing with some of the work that we did at the time, but that's all right, on obstructive sleep apnea. So what he did, though, is his project was to determine whether we could recover the signals. So he used a technique called blind source separation. And that technique is famous because it's widely used. The idea is you have 10 people in a room and there are 10 microphones. Each microphone picks out part of every person signal, right? Then you have these 10 signals. Can I recover the individual sources to figure out what every person is saying? So the microphone pick out mixed signals, right? And the problem is to recover each source. The problem is similar here because every contact picks out mixed signals from all different fascicles. And can I recover individual signals? So here's an example of what happened with this idea. We had 4 sources and 6 contacts. So here's one source, another source. You notice that these sources are overlapping by design, right? So we do the record the signals, and every contact sees a signal that looks like this. You can't tell anything about these actual sources just looking at this. These are clearly mixed signals, and you cannot pick out anything. But if you apply this blind source separation algorithm, which happens to be an ICA technique, in this case, you could see now 6 signals, 1, 2, 3, 4, 5, 6. Out of these 6, you have to figure out which one are the sources. You can clearly identify this one here, but this one doesn't exist, right? And this one doesn't exist either. So there are problems with this technique because it produces more signals than there are. And also, this method works on detecting, recovering signals based on their independence. You know, if signal A is independent of B, therefore, I can pick them out. There's one inherent problem there for us, because if you have one contact that's really, really noisy, noise, by definition, would be independent of other signals. So it'll look like a source. So it can work, and here's some examples of some results. We are looking at the cross-correlation. We've been able to come up with algorithms to remove the signals that were not supposed to be in there, and actually there was some pretty novel work there that Testa Yifus did. Then if you recover the signals and look at the cross-correlation of that noisy signal on the output from the noisy signal on the input, they were actually quite high. And here's this map here shows a major advantage of this technique. This technique is independent of the amplitude of the signals because it normalizes everything. And so that means that it doesn't really matter where the electrodes are. If I have 4, you can see I have, in this case, I have 4 sources, right? And 4 electrodes is not going to make it. But 5 electrodes is sufficient to recover all 4 signals pretty accurately with a cross-correlation coefficient about 0.9. And it doesn't matter where they are, pretty much. In fact, I'm not sure if I have the next plot here. No, I don't have it. But there's one plot where all the contacts are one end of the cuff, and you can still recover the signals. So that's a major advantage. But the disadvantage is this independence. That's a serial problem because if, as you saw, right, a nerve may have 3 fascicles going to the same muscle, that means that they're clearly not independent, and it may not be so easy to recover. So we then looked at different techniques, and this is the work of Brian Woodlinger. You guys know him. He just defended his PhD 2 months ago, I think. He's now in Pittsburgh doing a postdoc. Use a different technique. It's called beamforming algorithm. This beamforming is a weird name, but basically it's based on antenna design. So the idea is I want to receive a signal from Michigan, say, and I have a phased array antenna system where I got a bunch of arrays. Can I put some weights on one antenna and not so much on the other one to optimize the field of reception? So where the maximum or the best way I can receive from Michigan and not from Kansas City, right? We don't want anything from Kansas City. We want Michigan. So you get the idea, right? It's a bunch of weights associated with each receptor that will maximize the reception beams. And so that idea is applied to this because if I have a contact here and a contact there and a contact there, I can assign a weight to each one of them to maximize the reception from this little pixel there, or this pixel, or that pixel. So we can build maps for each contact to be specifically recording the signal from a particular pixel. And in principle, we should be able to build a map of the activity inside this fascicle. And you can see an example of that here. So again, we go back to the computer, did some simulation, see how this thing would work, and then went to the experiments after that. So we did some, Brian did some extensive modeling on this problem. And here's an example of some of the results he found in the 5% noise and for different sources at different locations within the nerve for a single source. So if I have a single source, you can see that in green is the detected area. That green, actually, you see is the center of mass of this blob here. That blob is where the source is most likely to be. And the green arrow is the center of mass of that blob. And that blob, determined from the beamforming algorithm, matches almost exactly with where the sources are. So this is sort of encouraging, but it's only one single source. When we have multiple sources, it's a little bit different. In any case, you can see that the recovery of that signal is quite good, because if you apply this technique, recover the signal for a bunch of time, and we're talking stochastic signals, we're not talking deterministic signals, right? You can see that the cross-correlation coefficient is quite high, 0.5. And again, see, again, that number here, 1.5 millimeters I mentioned earlier, same number. If I have separation of 1.5, I'm pretty good. We can do some good recovery. So this sort of matches with the work that Paul, you did on much simpler signals. So then we applied this, we wanted to try this idea on in experiments, and there, situation is not so easy, because to generate these signals, the real neural physiological signals, one idea would be to, say, generate sensation in the foot, for example, right? And I record from a nerve, and I see the stochastic activity passing through. It turns out that that's not so easy, because those signals are determined not just by the stimulation, but by the properties of the receptors. They're not stationary. It's difficult. We could have stimulated evoked signals, but then these are not physiological either. So what we did is come up with a different way of doing this experiment. And what you see there, I think, is the first time that it's been done in the peripheral nerve. This technique is to generate activity in the nerve using high-frequency sinusoidal signals. I know you're used to thinking of high-frequency signal sinusoidal can block axons, right? That's what Kevin's work's been done. But if you turn down the current a little bit at 10, 20 kilohertz, what you do is you enhance the activity of the signal. And this is what you see there. So this is a nerve, an ENG, right, recorded on the sciatic nerve of a rabbit with 100 microamp sinusoidal 20 kilohertz applied to the nerve. This is 420, and this is 600 and something 60. I can't quite see that. The sinusoidal 10 kilohertz is easy to remove because it's so high in the range, right? So what this technique is not new. We didn't invent that. It's Rubinstein has already applied it in cochlear prosthesis. The idea is to stimulate a nerve in a way that it will produce a physiological-like signal. If you stimulate with a pulse, you get an evoked signal, so compound action potential, synchronous activity of axons. But that's not the way nerves work normally. They work stochastically. And so in order to do this technique actually works quite well. So now what we have here is a way of controlling activity in the nerve, because I can apply the stimulus, the the 10 kilohertz, whenever I want to produce the signal. So we use this technique to generate signals. Then we apply these signals to the fascicles, 3 fascicles of the sciatic, and recorded the signal right there and try the separation using filtering and data acquisition and separation. And here's the experiment, and here's the first step in the recovery, and that is localization. In principle, I should be able to map where this activity is. And you can see here, these 2 fascicles, these are the blobs I was talking about, right? And they are plotted on top of the cross-section of the nerve afterwards. You can see it matches pretty well with this fascicle. It matches well with this fascicle. Same here, although there's a little displacement there. And same here, not bad here, pretty good. So even though these 2 are quite close together, you can still distinguish the location of the source. So the first step is looking good. We can map this activity, and in this case, we were able to actually do it with both fascicles active at the same time. And you can see that the results here. You see that one tibial and peroneal were both active at the same time. This is what you see on all the channels. You see mixed signals, you don't know what's what. But when you separate them out, you can recover activity in each one of them. Now, the nice thing, which is encouraging here, is that even though the signals are pretty low noise, we were able to classify them and show that the signal size depends proportionally on the current. So it's not a all or none side of detection. The signal size increases and can be proportionally recovered. So this is, you can see this is one fascicle, the other one is clearly not detected. So this shows that we can recover signals are mixed and that there might be a proportionality effect, even though, although I should say, that number is quite small. So the dynamic range of the signals is very small. And that could be a problem in the end. But at least within this range, clearly there is some linearity going on, and we may be able to recover them proportionally to produce controlled signals for a prosthetic arm. And this is where my next topic is. So I'm almost done. So we, as you know, DARPA was very interested in this topic, and there was a BAA for a reliable interface. Reliable interface in the peripheral nervous system. Jack Judy was in charge of this, and his big idea was, yes, everybody and their grandmother are working on neural interfaces. Everybody does it in their lab. Everybody has different technique. But there is no reliable demonstration that this can actually be done. Can we put an interface on a nerve, with a nerve, and show that day after day, I can get signals, I can recover them, so that these signals can be used for control? So I think it's a great idea, and I really agree with this idea, because he's right. We keep messing around with our labs here and producing some results on like 5, 10 animals, but that really doesn't prove the point. So this is what's been done already. This is what you want. You would like to have 10 degrees of freedom, 22 degrees of freedom actually, because that's what it takes to control an artificial arm. And you would like to be very low invasiveness. So this is where we love to be. This is what's out there right now. This is a 2 degree of freedom EMG, surface EMG control arm. And so it's non-invasive, so it's very low, but number of degrees of freedom is quite low. And this gap is a big problem. There are other techniques. This targeted muscle reinnervation here is a much more surgically invasive, because it requires taking nerves and then moving them in different places, different muscles, and reinnervating these muscles. And only 2 degrees of freedom, as far as I know, have been achieved with this technique. Another technique is the Utah array cortical models. And there, only 3 degrees of freedom have been achieved. And I would say that's just highly invasive, because that's right into the brain. So this is where we are now. It's not so good, right? Between here and there, there's a long way to go. What we're proposing to DARPA is a system that will be sitting here, about 12 degrees of freedom. It uses an electrode that I'll show you in a second. We think we can do 12 because we know we can do 2 degrees of freedom per nerve minimum from our experiments, so maybe 3. Since there are 4 nerves in the arm, that's 12. So it's pushing it, but I think it's possible. So here's the idea of what we're proposing. This is a proposal that I'm working on with Dustin, Pedram, who's here, and Bob Kirsch. The idea is this. We would place on an arm 4 electrodes that control from the main 4 nerves for controlling an artificial limb. And these 4 electrodes are connected to a device that contains an electrode and that has embedded electronics in it. And this electrode would have about 32 contacts. And the contacts would be multiplexed so that only 4 wires are coming out. And then these 4 wires would go on the surface of the skin where there would be another device connected there for interfacing. So this electronics package would be sealed, hermetically sealed, in titanium, even though it's very small. It can be done. It's on a difficult, it's a difficult thing to do, but it can be done, I think. So this would be definitely implantable. Then this would be, the signals would then be fed to the algorithm I just mentioned to you that would pick out which fascicle is activated. Then these signals would be fed into a controller that just have a little neural net here. And then this controller would then be fed into, the signals would be fed into the artificial arm. Now, in principle, this idea is that we can then control the movement of the wrist, fingers, and elbow. So depending on where you are, this platform would be embedded in either all 4 or 3 or 2 of these nerves. It's not so clear, it depends on the patient in question. We designed it in such a way that this device contains both amplifiers and stimulators. So that, even though we didn't propose that to DARPA because there was too much to do just right here, but they are also interested in sensory substitution. And so if we have stimulators here, we can activate fascicles selectively the way that I showed you. So in principle, this device placed on a nerve, if it does what we hope it will do, it should be able to pick out signals to robustly control this and also activate fascicles for sensory substitution. Here's an example of what this might look like. This actually is real. It's been built and tested. This is the work that Zengler Manrat did, was working in my lab. It showed the feasibility of taking a nerve and putting enough electronics on it to reduce the number of wires from 32 to 4. So this device has 4 wires and it can control 32 channels. However, it's been built, right, and tested. You can see the cuff right there. But this device was made of 3-layer copper flexible PC boards. And that's definitely a no-no, right, because you can't put that in the body. The techniques, there's no encapsulation known, I think, to man right now that can take care of flexible substrate without a hard box, titanium or ceramic. Although I think Second Sight has designed something that might be available pretty soon. But you can see this device, this device has 16 contacts on top, 16 contacts on the bottom, and there's some electronics placed on both sides, and you fold it, and it comes to this. The nerve is placed inside. And inside you have multiplexers, some counters for the addresses. We use the power supply line to carry information about the supply, the channel, and channel number, the waveform, etc. So there's some interesting design there that allows us to reduce the wires. And finally, just want to show you this. This is the latest piece of work that Brian did before he left. We're trying to figure out how this will look in the human. And so this is actually the sciatic nerve, cross-section of a sciatic nerve of a human on the computer. And what you see here is how the algorithm that I described to you could, perhaps in a real situation, pick out these different fascicles. In fact, in this case, we're not going after individual fascicles. The point here is that some of these fascicles are synergistic or carry similar information. So all the brown ones are in one group, the red ones are in another group, etc., the blue ones and the purple ones. So we put the signals, like I mentioned to you earlier, and you can see the contacts here are producing this. You can't see it, I mean, it's mixed signals. There's hardly any information available. But if you now apply the beamforming algorithm to this signal, to all 32 channels here, right? 32, you can recover pretty well the signals passing through these groups of fascicles. So in blue is the real signal, and in green is the recovered signal. So they all work except perhaps this one is not quite working quite well, but most of them are producing pretty reliable signals. So this shows that even though we may not be able to recover individual fascicles, it may be possible to recover groups of fascicles and produce signals that's sort of robust in time. But this is going fast, right? I mean, you need control within 100 milliseconds. You need to have at least a point to produce a movement with a frequency response of about 3, 4, 5, maybe 6 hertz pushing it. So that is something that might be feasible. And I'm going to stop here with some conclusions. So what told you today is all about the fine. And we'll be focusing on this design because we think it has a lot of potential. And it has been shown to be safe. I didn't show you any data on that, but I've done some experiments on that, and Dustin's done a lot of experiments on that as well. It's been approved. We have an IDE now for doing the experiments in humans. So this is going somewhere. The controller, I think, idea is working and could be used, but could also be improved. The beamforming algorithm has a lot of potential. We're now working with the mathematics department to improve the algorithm significantly. And so I think the conclusion is that this idea is probably a minimally invasive method for restoring sensation in patients, but also detecting movement intent and controlling the prosthetic device. I just want to thank people in the lab that worked on this, Wendy and Anjo Park, and I told you, with mathematics department, and Neil Clavetti is an expert on this, Dustin Tyler, Paul Hughes, Engleth Manrat, and Brian Willinger. And funding has been steady from NINDS and to support this project. And also we've got quite a bit of funding from the Department of Education for a training grant in neuroengineering. And thank you for your attention.
So questions for Dominique? Either speak up very loudly or use the microphone, please, for our guests that are online.
Durand, that was an excellent talk. My question was, you were talking about picking up signals from the nerves of amputees. So is there proof that the nerve signals in amputees truly reflect their movement intent? And the other is,
yes.
the sensory representation changes in an amputee, right? So when you try to feed back the sensation, you're assuming that the sensation in cortical respiration is still the same. Is there proof for that too?
Well, I heard the first question in 2 ways. We want, one aspect of the problem is to recover intent, that is motor signals coming down. So we want to pick out those motor signals to drive a artificial limb to produce a function that the patient or the amputee wants. Those signals, I think, should be more or less intact, although I've heard there is some demyelination, there is some loss of fibers, and to what extent that is still there, I'm not sure. I think mostly it's there. I'm hoping mostly it's there. Now, the other part you were asking the other way around, right? Is the stimulation you put in the nerve producing the same percept in the cortex, in the patient? That answer, that I don't know. And I think you might be right. There might be some significant changes in the way the plasticity of the cortex has adapted. But listen, I think that I can picture a time, maybe 50 years from now, where we're really good at this. And you have an amputation, and you place this device right away. Within a short time. So perhaps, plasticity won't have time to play a big role, and the patient will be good as new. We can always dream, right?
Thank you.
Yes, Ron.
Hold on, Ron. Hold on.
Dominique, what are the barriers to applying the signal processing and intent recognition techniques that you showed in the periphery to cortical signals? Are there barriers to that, or can this be transferred?
Yes, it can be transferred. In fact, Brian, who worked out this problem right here, is now in Pittsburgh working with a group that does exactly that for ECOG. He's not working on individual unit recordings, working on ECOG. So he's going to apply the same technique. Although I've heard, you guys heard when, I can't remember, I think Cynthia came give a talk on this recently, that on these units, it doesn't really matter what the processing is. All you have to do really get the power. And that's the only thing that matters. She made a pretty good case of that, even though I'm pretty sure it's controversial and a lot of people think there are some very specific statistical algorithms that are required to extract the information. But I think she's got a point. Maybe it's not quite as important as people think, and simple algorithm might just work just as well. But this technique can be applied, although I think we've been thinking about this to some extent. For some reason, this is particularly useful here because of the constrained nature of the sources. They are embedded into a cuff, and we have, so we take advantage of several things. One is a priori knowledge of the size of the electrode and where the sources might be. And that is crucial because that helps us build a matrix originally based just purely on that knowledge to do the mapping. You can't do this in the cortex because the sources are at different places. You don't have the a priori information. So now we're using also information about so once we've done the mapping, I didn't show you that, but there's another level of algorithm that takes into account the first level, which is the mapping. So I know this is a source here, there's a source there, right? How do I minimize the crosstalk between them? So we have an algorithm that sort of enhances the contrast between each one of those. So that we take a priori knowledge of where the sources are. And then there's still another level we're working on, which is noise, because we know what the noise looks like. And we can take advantage of that to further enhance the detection. And that algorithm actually based on an idea from another student of mine who's been a long time, Sri Nagarajan. I don't know if you guys remember him. He's now the director of the MEG Center in San Francisco. And he's doing source reconstruction in the cortex using that technique. Yeah.
I wonder if you could speak a little bit more about modality specificity in terms of fiber size. And most of these signals are going to be swamped by the larger amplitudes and the larger fibers. Have you actually, and you mentioned something about that it's that you looked at amplitudes, but have you actually tested whether you can specifically isolate smaller fibers and different I was asking about.
I mean, somebody up there in the front that could speak.
Yeah, the question is about diameter specificity. How do we pick out certain fiber diameters? So there are two problems. I don't know which one you're referring to. Are you talking about stimulation or recording?
Well, recording specifically.
Okay, recording, I think we try to get a larger signal as possible right now because signals are really small. So about 10 microvolts or something. And signal-noise ratio of these amplifiers is a problem because it's hard to make a really small amplifier that has very, very low noise. So we're taking our electrodes as long as possible. And these long electrodes are most sensitive to large fibers. Is there a way to pick out small fibers? Maybe, but I'm not sure I want to because then the signals can be much lower. And because right now we're not really capable of picking out the signals accurately as it is, I'm going to stick to as much as I get. So get it, if it's large fibers, well, let it be. Right now, we're working on other areas like C fibers, for example, trying to record these fibers, and that's really, really hard. They produce hardly any signal at all. And so small fibers would be nice, but I'm not sure we have this luxury right now. But if you change the length of the cuff, you can sort of shift towards smaller fibers and ignore the larger ones, but then the signal drops.
You were using still artificial way to produce the signal, to induce the signal. So what are your ideas before going to humans? Probably it's a good idea to have an experimental paradigm where you have a more physiological signal. So you stimulate the nerve, but actually it's the endogenous nerve activity where you are picking up. What are your ideas for the future for that kind of experimentation?
Yeah, well, we basically, one reason we did that is because we wanted a way to control the duration of the signal. So we can say, okay, now turn it on, turn it off, so we know. And then the second is that we wanted to improve signal-noise ratio because our amplifiers are not that good. In a real human situation, we are in the process of designing better amplifiers with the noise down to, this is what I told the specs for the next big grant, 0.5 microvolt RMS. So this is very low noise. I don't know if they can be achieved. The reason is that the electrodes are small because we have so many of them now, right? Electrodes are very small size, so they have big noise on their own. Just the electrode itself is noisy. So whether we can actually achieve that noise is going to be a problem. So we hope if we do get that right, then I think the natural signals in human will be sufficient. And the reason I am confident this is that we have done this already. So we have another student I didn't mention here, Masoud Sahin, recorded from cats on a whole nerve, not a 32 contact, a whole nerve, about a year and a half. It was getting natural signals reliably for that long. And clearly you could see, it was on the hypoglossal nerve, you could see the thing turning on and off for a year and a half. So it can be done, but he had external amplifiers that were very low noise because his electrode impedance was low. So it can be, but I don't have all the pieces right now to do this yet. It's a little frustrating, actually.
The stimulation side, stimulation side of things, you mentioned that redundancy was a problem. I was wondering if that's been explored as an opportunity, perhaps, for reducing fatigue in stimulating. If you could get the same output for two different stimulations, you could potentially have less fatigue in the system. What are your thoughts?
Good point. No, I haven't thought of that. But maybe, I don't know if other people have. To some extent, yes, I guess it has been done because when you put a large number of contacts, you can, I think there's a group in Vienna that did some of that work. You can switch contacts to produce the same response. But that requires a large number of contacts and probably more than we have because there are only so many fascicles, right, around that contact that you can activate reliably. So yes, I think to some extent it can be done, but there are also other techniques for reducing fatigue. And actually, you could argue that one of them is this high-frequency business, because in that case, the fibers are not continuously firing. There's a paper that just came out recently on optogenetics and activation of peripheral nerves in Taiwan mice with light. And light, these fibers respond to light because they have channel-rhodopsin in them, and they are actually activated. The small fibers are activated first, which is reversing their reverse recruitment order, right? And the system doesn't fatigue as much. And you could see that the activation is more stochastic than an evoked potential. So perhaps there's some hope that that can be achieved as well.
Okay. One last question here, Ron.
I have a question and a comment. I just think we need to get Lee Fisher on the NEC schedule for Friday mornings because he's using multiple contacts in his dissertation to try to reduce fatigue. But my question for Dominique, two quick ones.
Sure.
One, you mentioned a high-frequency technique for inducing neural activation to test your recording, and you just alluded to using it for stimulation, so I was hoping you could elaborate on that a little bit more. And secondly, in your DARPA proposal, you had this elegant idea of the electronics on the cuff to recover signals with 4 leads going somewhere. And I'm wondering what the plan was for those 4 leads between them and the prosthesis.
We have a plan.
That's to be determined.
We have a plan. No, we have a plan, and we have an exact plan. In fact, originally in the proposal, we wanted to implement that. It turned out to be too much money, and besides, they're not interested in that because the technology is already at hand. So what happened to the four leads, right? They go to on the inside surface of the skin that has a small titanium can, 4 connectors standard, right? And or ceramic, whatever. That contains a microprocessor to drive and process a signal that generates power. But eventually, I think that we would just scrap all that and plug it right into the Neuroprosthetics device that Hunter has been working on as the integral part of the system. And so there's a backbone already in place for supplying and providing signals. We just plug into that, and if it works, that would be another possibly interesting addition to that project.
And can this high frequency technique be used for motor activation?
Oh yeah. Yeah, I mean, you stimulate. We could see the muscle firing when you do that. It was obvious. But it's not. It's high power, right? Because it's high frequency. So, but you only apply it for a short time. So. It was used in a cochlear prosthesis a lot. It's still used, I think. Although some patients prefer it, not all patients like it necessarily. But there's a famous book on that where one patient that had a cochlear prosthesis was trying to improve his listening of music. It was Bolero in particular was the piece of music he was concentrating on. So he wrote this book. I don't know if you guys read it. I can't remember the title of it. He went to a bunch of places where they could try to improve the cochlear prosthesis design, and he ended up in ALA, Advanced Bionics, where they were implementing this high frequency to produce stochastic firing of the auditory nerve. And it improved a little bit. I don't think he didn't say, okay, now I can hear music, right? He didn't say that, but it improved a little bit.
Okay, before we thank our speaker, as a token of our
thanks.
appreciation.
I always wonder what's in those boxes, you know?